So thank you for inviting me to share with you my experiences. But I was more biased, so I decided to share my experience, what I'm working on. But I'll just briefly mention who I am and where I've come from. So I think, let me see. Um, I'll just mention where I come from and what I'm doing currently. So I did my my undergraduate statistics degree in Zimbabwe in statistics, mostly focusing on mathematical statistics. So I'll just mention briefly. So I did my bachelor's degree in Zimbabwe, where I was focusing on more mathematical statistics. And after a year, between the year when I finished my bachelor's, I did a bit of few things. I did a sentinel site surveillance, like poverty assessment in my country, where I was leading the province of Manicaland, where I come from, in assessing the poverty levels there with the UNESCO and also the Ministry of Social Welfare. And after that, I went into investment. So I became a money market dealer for whilst I was waiting to come to Hasselt after I got my scholarship. So after that, I went on to do an applied statistics where I did my thesis with Helena Gage, which had something to do with comparison of schizophrenia. And after that, I also did a master in biostatistics where I had a thesis with Professor Neil Hens and Professor Nicholas Paybrook, who was at that time at the World Health Organization. And after that, I decided to follow Emmanuel Lesaffre to KULeuven. And of course, I changed my color to be a Bayesian from being a frequentist. So I ended up finishing my PhD in 2011. And I decided to also go to Karolinska to pursue, to continue my research with register-based databases. Then in 2012, I decided to come back to Belgium in joining Janssen Pharmaceutica, where I entered the non-clinical statistics world, which I'll talk about today. And I've been in the what we call the manufacturing part of the statistics world. And the last year in July, I decided to switch to go to discovery. So at the moment, I'm an associate director in what we call TMEDS, where I'm focusing on discovery and non-clinical safety statistics. So that's in brief who I am. And if there are people interested to go into more details, what I did and the publications, you can just go to my LinkedIn page. And of course, also we can always network so that you understand what we are doing. That I can share also what you can do to get into the world that I'll be talking about. So that's about myself. So I think, let me see, I need to share this. So today I'll talk to you about non-clinical statistics world. I will just briefly touch on some example of what we do and some opportunities that you might find interesting, because most of the time I believe when we talk about pharmaceutical statistics, we focus mostly on clinical statistics. So the idea behind the pharmaceutical development pipeline is that we want to design products which are of a good quality, and also that the manufacturing line itself is not interrupted, that it gives us good product, but also it's consistent. I think these days you can understand with COVID that when there's a recall, it's really catastrophic. A catastrophe. You can think of India at the moment, that if, let's say, Janssen product is recalled or a Pfizer product is recalled, it really is a needed drug and it can really cause havoc on the market. So one thing we do is we try at least to account for all the risks as we go through the pharmaceutical pipeline and see what risks can be carried forward and what risks have to be mitigated. So at the end of the day, all these pharmaceutical development studies have to be based on the quality risk management. And what do I mean by that? This is actually an interdisciplinary team of experts where we bring people with knowledge of chemistry, knowledge of biology, engineers, and also statisticians. Then we want to quantify risk, not only from the perspective of chemistry, but also from the perspective of even the financial part of the company. That at the end of the day, we want to maintain the company to be able to produce medicine, but also balancing that with the consumer risk, that is the health of the patient. So essentially, statisticians are there to quantify risk and to bring an understanding to the sources of variability at every stage of pharmaceutical development, the research and development pipeline. So that's what we do. We deal with variability, we deal with risk, and making sure that these risk concepts are understood in the various disciplines in the company. And also because we need also to go to the authorities, the FDA, the EMEA, to present this statement in a language which is understandable, not a statistical language where we deal with betas and alphas, but a language which the everyday man can understand. So when I talk about the pharmaceutical development pipeline, this is what I'm talking about. We start with the preclinical studies. Here we are looking at the chemists and the biologists thinking up of a molecule and how this molecule work. And hopefully this molecule work well in animal models. And after we found molecules which work, we now need to package them to be a component which goes into a human in a clinical trial after having filed what we've discovered with the FDA. That's why we make our tablets. That's why we add excipients like to make the tablet test well, to make the tablet be absorbed, and all sorts of things. And from there, we go on to this phase 1, phase 3, where the clinical statisticians will deal with that. Then finally, we file our vaccine, we file our PU, and then it's approved by the authorities, and then we have it on the market for the patients to alleviate their pain or to get better in their situation. So when we look at this, there is a divide in the pharmaceutical world. We have what we call non-clinical statistics. Usually, this is statistics which applies to or is limited to things which do not involve humans. But you find that this is just a cutoff which is there in general, but you find that non-clinical statisticians can also enter into the clinical world also because it's all linked, it's one pipeline. Then we have also clinical statisticians, and these are normally the statisticians in clinical trial supporting things which are needed by the FDA. And in the non-clinical statistics area, we can divide it into 3 parts. We have discovery, where we work with biologists who are looking at the molecule which come from the chemists to see how it works, how it can link with within the omics setup with certain genes, and how it can shut down certain receptors on certain cells when it comes to oncology. And we also have non-clinical drug safety, where we look at, we have found an efficacious drug, but then can it be tolerated by a patient? So we have to apply this also in animal model at the earlier stage. Then we come to the chemistry, manufacture, and control, where we are looking at things like expiry date. Does it dissolve well? So when you get recommendation from your doctor, like you take the tablet 1 time a day, you take the tablet 3 times a day, it all comes from statistics. So behind all these things like the expiry date and the dosage, and also the dosing range of a tablet through the day, it is statistics involved in that. So I'll discuss that briefly to give you some examples. So I'll talk about non-clinical, preclinical studies in this. I'll not talk about clinical. I think for clinical, even in statistical courses, you get to learn a lot about that. So I'll talk about discovery, which is in red here, and I'll talk about manufacturing, which is in blue. For my work in Janssen, I've been from 2012 till last year, I've been in the blue area here, where I'm supporting manufacturing. But now I move to see where the actions start in the discovery part. So discovery studies is concerned with actually you want to identify the chemicals which you can use to fight a disease. And after you discover those chemicals, you engage biologists to now see the mode of action of those chemicals. So that at the end of the day, when you go to the authority like the FDA or the EMEA, you really understand what you're proposing as a new molecular entity that you want to push in your pipeline. And there are skills involved which are general. I just put some general skills. But the first thing is communication. As I told you that the risk management is managed by an interdisciplinary team. So you need to be multilingual. When I say multilingual, I'm not talking about speaking French or Japanese. I mean speaking the language of chemists, biologists, or even of business people. So at the end of the day, you need, like what was presented about funding, you need to be able to embrace your partners and know what they are talking about, and know there is even reservation when you come to propose them, let's say, a p-value or a confidence interval. So you need to be able to adapt to their, so that they can learn from you also. So it's a give and take mentality. So we find that in discovery, it's a lot frequently statistics use, but also we are trying to drive some Bayesian statistics when it comes to certain aspects which are easy to calculate using Bayesian statistics. Then we have also omics data, which comes into play. So we need also not only to learn biostatistics, but also to learn the bioinformatics, statistical bioinformatics, and even epidemiology. So also of interest is software skills, which you can use to help you to not always to be there to push the button for scientists, but to allow them to be part of the process when it comes to statistical analysis, where they can run things on shiny applications. Then also another part is to train them to have good design of experiment so that they can easily disentangle variabilities, disentangle the effects that they want to look at. I will not talk about toxicology because of the time limitation, but however, in discovery, you're trying to find how effective your molecule is, but in toxicology, you're trying to play also to the safety of the patient. So in discovery, I decided to give you some examples. So in discovery, normally in oncology, where we are dealing with cancer, we know that cancer cases is hard to treat, and there are no clear medication that, or we can say the golden pill for cancer. So we find drug A is effective for, let's say, a cancer, drug B. So what we try to do, in what we call combination of synergistic studies, is that we try to bring these two drugs together. And we hope, like in this diagram, that we have concentration of A and concentration of B. If you combine them, you'll find that your cancer cell, we can say your cancer are killed more. So you expect this to come from the diagonal to go down so that you have a bigger effect. But if it's actually lowering the effect, it becomes antagonistic. So we find something like this. We can have a plot where we have a surface, and if your points for the combinations are below the surface, you have a good or a synergistic drug combination. If they are above, it's really becoming even less beneficial. And I think I'll click here and to just show you something. Let me see. Let me come out of. So this is an example of such a combination. We have two drugs, A and B. We have the dose effect here. And when you combine them, you get another plot, which I'll show you. Oh, let me see. It's supposed to be here. So I don't know why it's not plotting. But anyway, don't worry about it, I think. So basically, the plot will be like this, where you can see in two-dimensional if you have a synergistic effect or an antagonistic effect, or it's nothing, it's just additive. You add the two drugs and they go to different site of action and they continue to work. Then before I go to the next one, what we are trying to do also, which is a current project in which we are dealing with, is to try and have an economic selection of candidate combination, because when we do these synergistic studies, we might have 20 drugs to combine to have 2x2 combination. But then if you are going to do it, you are going to spend a lot of resources, where 70% of those combinations are not going to be effective. So we have tried to embark on putting a design of experiment to already filter through the non-significant synergistic effect before we go to invest a lot even to do the full range of the dose response for drug A and drug B. So that's something we are looking at also, to use design of experiment to cut back on the costs of developing drugs. Then we know that when we are in discovery, we also do experiments on mice, for instance, or on animals. And normally from ethical committees, we are not supposed to just collect 100 mice. So if you come in pharmaceutical and you talk about, I want an experiment, you cannot expect to get 1,000 mice. We are bound by ethical consideration to take a few. So, however, when we do that, we have a challenge in traditional randomization. So at the end of the day, we develop apps also to help scientists that they can randomize their own mice, a small number of mice, but accounting for all the prognostic factors. And we have an app called Irini app, which scientists can just use instead of us sitting down with them and doing an analysis to create an experiment every time. They are able to do that because we know what they want, and we have developed a tool which can visualize what they want. So in the interest of time also, I will not go deeper in the example, but I just want to mention that we talked about discovery, and discovery is looking at the new molecular entity. When you got the new molecular entity, you want now to make this chemical or this biology to make it able to be designed for a human. So we find that in CMC, there are a lot of things being done to understand the quality of our drugs before and when they are in clinical trials, so that we prevent unwanted effects on people. So we find that actually communication again is important, not only of the interdisciplinary team, but across the pipeline, because the discovery people have to feed the CMC people to be able to understand what is it they have, so that they can build a good profile for the drug. So here we find that Bayesian plays an important part. It's not in brackets now because it really plays an important part. You need to calculate the probability, the risk of even not getting the dosage which is intended for you. We have design of experiment because now, unlike in discovery where it's less regulated, in CMC you have to supply documents to the authorities. So everything have to be above board. You have to check quality. You have to check what you are writing. Is it statistically sound? Is it biological or chemically sound? And we also help in having our markdown reports for scientists that we can easily change and adjust things when needed, even for the reports for regulators. And just to give you an example, so when you are assessing quality of, let's say, a drug, you are not going to look on one thing. You are going to look on various things, the ingredients itself in the drug. You are going to look at the excipients or let's say the sugar you want it to taste well, let's say for babies. And you are going to look at which packaging is good. And you are going to have measures, what we call measures like assay and dissolution, water content, how much water is in your drug, the color, its ability to disintegrate. So at the end of the day, we normally, when we want to create drugs, we put the drugs which we formulate into a stability chamber. What is a stability chamber? It's like this thing. It's not always looking like this. Where we look at the climatic zone in Africa, in Brazil, in US, in Asia, and also refrigerated condition. And these conditions are the conditions we see on this planet, so that we make sure that our drug is good when it's posted to Zimbabwe, when it's posted to France, when it's posted to Asia. So, and when, for example, you look at assay is just that the ingredient in the drug. If we say we have to have 5 milligrams, it should be, let's say 90% of that 5 milligrams should be there in that drug if we are to consider it to be effective. So here we monitor the drug for some time, for months, and we check where, if we put a prediction interval where it cuts the 90% line. So that will be to say our shelf life here, for instance, is 20 months here, because we know our drug for some of the batch is failing below the line at that time. So that's why we need Bayesian. That's why we need also to do mixed models and also to do fixed effects model. to come to an understanding of when we put our drug in the pharmacy, how are we going to gauge the time it is going to be effective for a patient? So that shelf life, consideration. Then we also have a specification setting where we look at which specification can we file with the authority. Then you all use also studies for that. And one thing which I wanted to share is that I'm talking about this stability monitoring. Normally, we know our drugs normally can take 2 years, but you cannot go to the clinic before you file for some form of specification. So you cannot wait for 2 years. You lose money. Your drug will be late on the market. Like, for instance, if you look at the vaccines that we are having, you cannot wait too long because it's needed. So what we normally do, we do what is called accelerated predictive stability testing, where we stress test these drugs. So from an equation by Arenas, Swande Arenas, who linked degradation rate to temperature, we extended that to include humidity, because when you look at temperature conditions, which I have showed you, there is temperature and humidity, and these two things affect the durability of drugs. And I'll not talk much about how you get it, but it's all statistical derivation and linear mixed model, and you can do it in various ways to get to let's say, some shelf life.
Sorry to interrupt. Can we wrap up?
Okay.
In a minute? Thank you.
Yes. So the other part which I wanted to mention in CMC is also dissolution. What I talked about, that when we need to know the required percentage of the active ingredient to release in a given time. So if you have a headache, how much of the drug you need to take? So if it's what we call, let's say, a drug which can release the whole day, you can be given one drug. But if it's a drug which is immediately eliminated, we need to give in the morning, in the afternoon. So what the doctor tells you, it's actually coming from a statistical evaluation of how your tablet dissolves. So we can think of it as efficacy within a day. But we need also to maintain a consistent behavior across time. So at the end of the day, also we can model the stability of dissolution. So dissolution, I'm just giving the screen here. What it is all about: you dissolve a tablet and you monitor it. You use HPLC to measure the concentration, and you monitor it over time. And here I just give an example where you model the dissolution let's say within a day with this dissolution time using a beta regression model. And here I've just put the code of how you'd do it in R. Then to conclude, I just try to give you a glimpse of non-clinical statistical world, because most of the time, even in my time, I didn't know about this world. I only know about clinical statistics. So when I entered that world, it was interesting to me. And you find that for clinical, you are highly regulated, because in clinical trial, you're going to the authority, but you can have more fun in non-clinical statistics. You can try your models, even coming from an academic setting, of the things which you want to do. You are more flexible. DOE concepts are very much required in CMC statistics. So it's good to have a basic understanding of that. But also in discovery, we are also trying to promote that within the people, the chemists, to understand what is going on. So to paint the risk map at every stage, we need an interdisciplinary team of experts. And the non-clinical stage is pivotal in balancing competing risk narratives, because a risk to a chemist and the risk to a biologist are different, or a risk to we can say the shareholder. So we need to be able to balance all these things and to be able to speak their language. And of course, when we have this multilingual concept or languages in terms of our fields, we can have more fun, because we now understand the direct impact of what we're doing as statistician towards the other fields. And I just put some books if you want to know more. There's a book of non-clinical statistics which you can read. There is also a book of CMC and also some Bayesian concepts. So I hope I've given you a brief of how the world of non-clinical statistics looks. Sorry for speaking at an advanced stage, at an accelerated stage, but I hope to just share briefly what we do, and hope to see students also being able to come and collaborate with us. We usually invite ourselves like to Hasselt when they have the Masters Day, and also to Ghent when they have a Masters Day there to really look for research. We are always open to collaborating with academia, like we are always doing that to be able to also get more methodological development in non-clinical statistics. Thank you.